So, hello everyone, good afternoon, and welcome to today's episode of the ESDR Kitchen webinar. This is the webinar series of the ESDR, which we have been running for a couple of years now. Thank you very much for joining us today, even though it's in the middle of the summer. So this episode is part of the series, which is called the Recipe Book, where we can learn about new methods, which we can use in skin research. And it is my pleasure to chair today's webinar together with Professor Anna Balato from Naples, Italy. So today we will dive into skin cells and learn about one of the methods which was actually listed as one of the 7 technologies highlighted in Nature last year. So it seems like a very exciting technology. So our speaker today is Ross Lowes, who currently works at the Electron Microscopy Research Services at Newcastle University. And he has over 8 years of experience with microscopy and image analysis. And he's also studying for a PhD, developing bioimage analysis workflows. And he's working with my fellow board member, Professor Neil Rajan, to study cylindroma tumor cell morphology. So very welcome, Ross. The stage is yours.
Thank you for the introduction, Enikö. I'll just start sharing my screen. Okay, can everybody see that? So, to start, just to introduce the topic I'll be talking about, I'm going to be covering 3D bioimage analysis of volume electron microscope images of skin cells today. So just to give you an idea of what Volume EM can do, the video on the right-hand side is just a fly-through of some of the cylindroma tumor cell data that we are looking at in the cylindroma study. So this is just to give you an idea of what you can do with visualizations of the data. To give you an idea of what can actually be achieved, on the left-hand side here, you can see a nucleus of a cell surrounded by mitochondria. So Volume EM can allow you to visualize your data in 3D down to a subcellular and organelle level resolution, so that you can actually distinguish individual organelles in your data. And the other thing it allows you to do is to actually see structures that are impossible to see in 2D. So in this bottom image here, you can see that there's a 3D representation of the mitochondria, a 3D model, I should say, next to the actual data that it is from. But when you've got one slice, you can only see that the mitochondria look like basically round blobs or kind of sausage shapes like you'd find in a textbook. But in reality, the mitochondria have this interlinked, complicated scaffold-like structure in this particular sample here. So you wouldn't be able to really see that on a 2D image at all. It's impossible. So you need 3D visualization to be able to see that. So just to introduce you to some of the topics I'll be talking about in the presentation. To start, what is bioimage analysis? Well, bioimage analysis focuses on the quantitative measurement of biological systems in multidimensional image data, being as objective as possible in measuring features in bioimages. So this is from the NuBias website, which is the network of European bioimage analysts. And just to talk briefly about the disease that I'll be mentioning today and some of the data, which is cutaneous cylindroma. This is a mutation of the CYLD tumor suppressor gene, which is a deubiquitinase enzyme that removes ubiquitin from the proteins involved in regulating cell growth. This mutation results in uncontrolled cell growth, usually in the follicular cell compartment. And the tumors are also prevalent in follicle-rich areas as well, which is another indication that it's of follicular origin. Now, the other core topic I want to talk about is electron microscopy. So what is an electron microscope? Well, electron microscopes use electrons instead of light to illuminate a sample. There's two main types: a transmission electron microscope, which images through thin sections of tissue, and a scanning electron microscope, which images the surface, or in this case, the cut block face of the sample. The image on the right is just in a transmission electron microscope. So what is volume electron microscopy? Well, volume EM is more of a technique than a microscope. It involves the capture of large-scale 3D data sets in some kind of automated sectioning process. There's three main types, including SPF SEM, which I'll be talking about today, FIB-SEM, and array tomography. So the system we have is a SPF SEM, or serial block face SEM. So serial block face and FIB-SEM both look at a cut block face of a sample, and I'll explain that in more detail as I go on. So just to give you an idea, this is what the system looks like. So the 3-view system that we have is actually inside the machine under vacuum, because all electron microscopes, I should say, are under vacuum inside to be able to function properly. So the image you can see on the right-hand side is, just for context later on, the red circled area is where the knife is on the ultramicrotome. So this is a diamond knife used to cut the sample, and the green circle is the sample itself, with the block face being this little black square here. So that's the area we're actually looking at on the microscope. So how do you prepare samples for SPF SEM and then image them? So sample preparation involves fixation of the samples and then heavy metal staining, followed by dehydration into acetone, so it's not drying, and then resin embedding afterwards to polymerize the sample into a hard resin. When you've done that and polymerized your sample in resin, you have to cut it out of the resin and basically unearth it again. And then you take the fully polymerized sample and cut it off from the resin and mount it onto an aluminium pin. So the sample at this point is completely solid in the resin once it's polymerized. So all the way through, it's not just enrobed in it, it's actually penetrated into the tissue. So the imaging process involves basically you put the sample on the pin into the machine, and then the ultramicrotome in the machine, once you set the imaging away, cuts the sample, the surface of the sample, to reveal the cut block face. You'll already have a block face to start. That's how you find your imaging regions of interest. And then what it does is, after it's cut, it images. So to go back to the earlier image, you can see the diamond knife there, circled in red, is in that area. What it does is it moves across, and then it retracts and cuts a section off. And what it does is it repeats this process, steps 1 and 2, cutting and imaging as many times as you require. And what that results in is a stack of images, not just one image, but many images stacked on top of each other. This is routinely up to 400 slices, which could be about 40 microns in depth. And just to give you an idea of what that looks like, here's a video running through the slices one by one. So this might look like a time lapse of a cell growing and then disappearing, but what it actually is it's the machine cutting through the sample in the Z dimension. So how do we use bioimage analysis to analyze the serial block face SEM images of skin cells? Well, to start, what you have to do is post-process the data after it's been captured. Post-processing depends on the volume EM system that you've used, and post-processing of images can be automated using batch processing and macros in different software. So post-processing often includes things like filtering and then physical steps like cropping the data down to the region you're interested in, and things like alignment, which is to correct for erroneous movement in the data due to, say, beam shift or kind of the sample wobbling due to someone walking past the machine or something like that. So then what you can do is other steps like contrast normalization, which will help remove any linear or non-linear changes in contrast as you move down through Z in the data as well, which is the cutting dimension basically as you move through. So once you've done your post-processing, the next part is to segment your data. So segmentation is the process of dividing an image into regions of interest. And what that involves in the simplest way is basically doing manual segmentation, which is where you draw around the region of interest in your data. So this is sped up 16 times. So this is a very slow process. So ideally, you want to try and automate this to kind of speed it up. But on a lot of data, it's very difficult to do because there's not an amazing signal to noise ratio between the feature that you're interested in and the background in a lot of EM data. So there's some steps you can take to improve that depending on your data. What you can do is use a watershed tool, which looks for dark or light boundaries around an object, which cells and nuclei often have. And this is another sped up video just showing what it does is it divides the image into superpixels and allows you to select those superpixels with the brush. So you don't have to do as much work. But as you can see, there's some error at the bottom of the cell here. So you have to correct things like that manually still. But if you've got an object that has very nice dark borders around the outside, then it makes it a little easier. Like a nucleus here can be segmented in seconds using this process with only a small amount of manual correction at the end there just to clean it up. So what you get when you've done that is, for example, you'll have your labels for the cell membrane, I've got the basement membrane here, mitochondria, and altogether you get a label image that looks like this. And you would do this on all of the slices where your object of interest is present, like this cell in this case. And then what you can do is put them together into a 3D model, and the software can reconstruct that into a physical, like a 3D model that you can then look at and rotate in 3D. So segmentation generally always starts with manual segmentation. This is time consuming, but sometimes necessary if you want to generate what's called ground truth data for machine learning methods. Just to give you some context, ground truth data is seen as the expert standard for what the segmentation should look like. So based on what a trained operator sees when they look for, say, a nucleus or mitochondria in the data. There are some conventional automatic methods available, but they still capture erroneous parts of the image sometimes that you don't actually want, so you still have to correct a lot of that. But there's also these days AI-based methods that can speed up the process and sometimes be even better than the current conventional automatic methods. Just to give you some context, machine learning segmentation involves ground truth data generation by you, where you manually or semi-automatically segment the object that you're interested in the images. Then what you do is you then expose the model, which is the name for the kind of overall system that is trained to recognize the objects in your images. You expose that to the trained ground truth data, and it basically learns what you want segmented from the images. And then ideally, the output is the automated segmentation of the features that you want in your images. This, however, is way more complicated and time consuming than this charming little diagram implies. And it can help to have people that you know in bioimage analysis who can help you. And there's a lot of people out there who can. And I'll introduce some of the forums and things at the end that you can go on to talk to people about it. So machine learning segmentation, depending on the difficulty and the type of problem, different tools can be used to segment your particular object of interest. The field is rapidly developing, and there's some great software packages and tools out there, including the following. There's BioImage, Fiji, and DeepImageJ. DeepImageJ allows you to download models from the internet, pre-trained models that you can use to segment your feature of interest if it's been trained on that already. So for example, a mitochondria model, you might be able to then apply to your data as well if it's volume EM. Then you've got iElastic and deep learning for microscopy everywhere as well. So machine learning tools are becoming more accessible all the time, but they're still not very accessible to non-bioimage analysts. So it's good to be able to have people to kind of talk to in the field who can help with that. Once you have segmented your data, what you might want to then do is move on to measure and analyze the features that you've segmented. This is a bit word heavy, so I'm going to briefly cover these topics and you can ask me more at the end if you want. So many metrics can be calculated from the segmented data, including physical measurements such as volume, size, and surface area. You can do distances between objects, average distance between all of the objects as well to give you an idea of clustering. And then you can also look at dimensionless properties such as surface roughness and shape descriptors. Now, a shape descriptor is basically like sphericity, for example, which allows you to compare a 3D object to a perfect sphere. Like on the right-hand side, we have a perfect sphere, which has a sphericity of 1, and this cloudy blob-shaped object here, which has a sphericity of 0.45, for example. So the further away from 1 the number is, the further away from a perfect sphere it is. And this can give you an idea of how round all of the objects are in your image. These can all be calculated using the following software, such as Fiji, MIB, Amira, and Amoris. So what I'm going to do now is just give you a little kind of case study example of what I've been working on with Neil's data as an example of a bespoke bioimage analysis workflow. And this is to analyze the mitochondria position and distribution in skin and cylindroma tumor cells. So what we noticed when we looked at preliminary TEM images was that mitochondria appeared to distribute heavily to the basal side of the cells next to the basement membrane in cylindroma tumor images. Preliminary TEM analysis indicated that the mitochondria area in the TEM images was higher in the basal side of the cells, which is the pink mitochondria, than in the yellow mitochondria in the apical side. So we wondered if mitochondria could be playing some kind of metabolic role in the breakdown and deposition of the basement membrane during tumor growth. But also this could be applied to maybe more relevant organelles to this later on down the line once the technique works. So how can we use bioimage analysis to do this? Well, we actually had a master's student originally find a way to divide the cells in half to then be able to look at the mitochondria population in one half of the cell versus the other. But I wanted to try and make this a bit more reproducible and unbiased. So what I did was find a way to automatically split the cells in half based on their proximity to the basement membrane. And this gets rid of the human decision part from the workflow in this case. So step one is to measure the distance of the cell, the center of the cell to the basement membrane. Then step two, you dilate the basement membrane to this distance to give us the basal region of the cell. Then step three, co-localize the mitochondria to the, look at which mitochondria co-localize to the expanded region of the basement membrane. So the green area would be the basal side of the cell and the yellow, the apical region of the cell. And just to run through what that look was like in 3D. So this is the 3D reconstruction. You've got the basement membrane in pink, the mitochondria in multiple colors, and the cell in yellow there, the cell membrane. So if I expand the basement membrane to the central point of the cell to that distance, and then use that to co-localize which mitochondria are in the apical and basal half of the cell, then we get rid of that and the cell membrane, and what we're left with is the two populations of mitochondria divided up for us to then analyze. So what we can do now is export the results as a CSV file, and then post-process this output to determine the volume of mitochondria in each region of the cell. So what I've got here is just the example data from this. So we can now statistically analyze it and look at the apical mitochondria volume in the cells versus the basal mitochondria volume in the cells. And this is for all the cells we analyzed in the images from the study. But depending on the resolution and quality of the segmentation, you can actually analyze all of the metrics I mentioned earlier in each region of the cell. So not just volume in this case. So in summary, Volume EM can allow you to analyze 3D volumes of tissue at a nanoscale resolution. This allows for insights into the biology and pathology of the skin impossible with 2D imaging. And bioimage analysis of Volume EM data can provide even more information about your data as well. And just to finish, I'd just like to acknowledge Professor Enikö Rajan and Professor David Steele. They're my supervisors on my PhD project, and thank them for all their help and support. And my boss, Tracy Davey, as well. And also the donors of the tissue used in the cylindroma disease study as well. And I'm just going to leave this up during the Q&A unless I have to change slides, but I've just got a little bit here talking about some important links for bioimage analysis, including FAIR principles for bioimage analysis, Bioimage Model Zoo, where you can download pre-trained AI models to segment your data, and ImageSC, which is a really nice forum for bioimage analysis. And a lot of the developers of the software are on there for you to contact and talk to about problems with the software you're using. So if anybody wants to ask me any questions, I'll be around for the QA afterwards, and thank you very much for listening.
Many thanks, Ross, for this very interesting presentation. And
Thank
just to break the ice, I mean, I will start with a question to take a little bit of time and also inspiring participants to make their own questions. But I was wondering, I mean, this is a very fascinating technique. You showed us very nice images, but at the same time, it is very time consuming. You have to be very dedicated to this technique. And I was just wondering, in this ever faster and faster world, when researchers usually want to have the answer now, okay?
Yeah.
How do you think to attract young researchers to start to think to this technique?
Well, what we are hoping to do, and what I'm hoping to do after I finish my PhD, is actually develop a kind of fully automated workflow for processing the data in the facility that we can deploy to users who come in to look at specific kind of targets in the data. So, for example, if someone is looking at mitochondria, the plan is to be able to analyze their data sets and capture their data sets, then post-process the data automatically, and then segment the data after that automatically as well. So they can immediately then start to go on to get insights from their data in, say, less than like a week's time. So what we could then do potentially is capture a data set and have it post-processed and analyzed in like a matter of days, as opposed to at the moment it can take like weeks and if not more to get to the point where you've got insights from like a particular data set, because it takes so long to do the segmentation. But with some of the new deep learning workflows, we can more effectively train the AI models to segment out the features of interest with not an enormous amount of training data. So basically, that's the plan, is to bring people into the facility, is to make it so that we have a workflow from start to finish where they can go from not just get images out at the end, but get data out at the end as well that they can then publish alongside their other techniques that they've used. I hope that answers the question.
Yeah. Thank you.
Yeah, thank you. While we are still waiting for some questions, I may ask you, because we had a previous episode, it was some time ago, on cryo-electron microscopy, which is another technique which has people have been talking about because also of the Nobel Prize. So my question is, when would you use one when the other technique? Of course, both have their advantages, disadvantages, and their special areas of use.
Well, that's a really good question. So for cryo-EM, if you would use that often if you're looking at say small particles. So a lot of the time at the moment, people use it for reconstruction of proteins and things. So you're looking at stuff on the nanoscale, basically. And sometimes people use it for kind of cryo tomography, where they've got a thin section of the tissue and they'll look at the thin section in a tilt series, I think, generally, to get a 3D image of that. And also, sometimes, though, people will use it for tissue at a similar scale to what we would do volume EM. And it's often when they want to look at the tissue as close to its native state as possible, but you don't get as good contrast with it generally. It's quite hard to do. I can't really speak because we don't have a cryo FibSem system here, which would have the sample frozen and then milled away by what's called a focused ion beam gun. That's the other technique for volume EM. So I can't speak so much as to why people go for that as opposed to volume EM. But definitely for volume EM compared to, say, cryotomography, the scale's completely different. So that's one of the reasons. People do cryotomography just because they're looking at stuff that's on the nanoscale. Whereas for volume EM, you're looking at things on the kind of cellular scale. You're going to be able to capture much larger volumes of tissue much easier than you would for cryo. Even something like cryo FibSem, where you're still doing volume EM, but on a frozen sample, you're going to be able to only really image a small region of your tissue. Whereas people who do connectomics, so neuronal data analysis stuff, they will look at sometimes volumes of the block face that they're cutting away that are kind of over 100 microns or more in size, and then reconstruct all of the neuronal connections within that data, or try to anyway. So they can't look at those things in cryo-EM because the scale's just so much smaller. So it basically gives you a lot wider region of interest, but not necessarily as close to the native state of the tissue and cells.
Well, thank you. I think this is very good also for the audience to clarify this, and it's very exciting with all these new imaging techniques.
Good.
We are in perfect time, and I can see that up to now there are no questions from the audience. So,
but you may be available by email, right? If anyone thinks about the question afterwards.
Yes, I will just quickly. Will I have time to put my email in the chat, or do you want to share it with people afterwards? So I don't think I put it in the presentation.
We can share afterward. That's fine. Thank you very much.
Yeah.
Okay. Thanks very much.
Thank you. So I think we can conclude, and we have just a couple of announcements, no? Okay. So before to conclude, I'd like to first to thank you, Bristol Myers Squibb, to support this episode of the ESDR Kitchen, and to advise for the next episodes that will be on the 21st of August. You can see here, we'll have Camille Brown and Pio Maybe I didn't pronounce it well. very interesting points. We can go ahead. And of course, all the episodes are always available on YouTube, and you can find here on this link. And this is very important because I want to tell you all that still open the late breaking abstract submission. So there are 2 days left when where there is still the opportunity to present your abstract for the next annual ESDR meeting. That will be from the 4th to the 7th of September in Lisbon, Portugal. So we will meet, of course, in Lisbon. We will wait for you all.
And thank you all for listening today.
Bye-bye.
And thank you very much for your presentation.
Thank you, Ross,
Bye-bye.
really.
Thank you. Thanks for letting me talk. Goodbye.